Good afternoon, everyone. Welcome to our next in the series, specially targeted education program, Building COVID-19 Vaccine Confidence by Educating the Educators. Just to remind you that this program is being run in collaboration with the SickKids Learning Institute, the Black Creek Community Health Center, the Scarborough Health Network VaxFast Clinic, and the Black Physicians Association of Ontario. This afternoon, we are privileged to have our own Dr. Upton Allen. That doesn't require a lot of introduction to you, even you've been participating in the series. He's the head of the Division of Infectious Diseases at SickKids. And this afternoon, he'll be speaking to us about what's new with pediatric vaccines, third doses, boosters, and more. This session will be recorded as usual. And before we do that, let's do the land acknowledgement. We would like to begin by acknowledging the land on which SickKids operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Petun First Nations, the Seneca, and most recently the Mississaugas of the Credit Valley. Today, Toronto is home to Indigenous peoples from across Turtle Island. SickKids is committed to working toward new relationships that include First Nations, Inuit, and Métis people, and is grateful for the opportunity to share this land in caring for children and their families. Without much further ado, I invite Dr. Allen to do his presentation, and there will be questions at the end, so hold them. You'll be invited to post the questions during the discussion period. Dr. Allen, over to you.
Thank you very much, Isaac. I sincerely appreciate the opportunity to speak today. The outline of my presentation is shown on the slide, where I'll first do a quick overview, if you will, a bit of a recap on the available vaccine types that we have, and then focus on a question that I think we probably, from the point of view of the scientific community, could have done a better job articulating, I think, much earlier on, as this relates to what is it that we try to target with the COVID-19 vaccines. And then I'll address 2 or 3 lingering questions, these questions that never seem to go away, that keep popping up every now and then. And then I will then talk about the third doses, questions specifically, are third doses effective in the different age groups, and are third doses safe? And then we'll talk a bit about the mixing and matching of vaccines and choosing different types of boosters, and then we'll sum up. Let's start with the question of what is it that we really target when we talk about vaccines? What are vaccines really doing? What are we trying to prevent? And in general, we try to prevent not 1 or 2 or 3, sometimes 4 different outcomes. First, with any vaccine, if you have the vaccine trying to prevent a communicable disease, you want to ensure that the person who gets vaccinated is prevented from dying, or prevented from having severe disease, such as might end up putting them in hospitals, or more moderately severe disease. And then in an ideal world, you'd like to prevent any infection. And so those are the different grades or degrees of effectiveness, if you will. And so to think of examples, let's look at a few of these examples, and I'll come back to that later on. But we will focus on examples of vaccines that have actually prevented different types of illnesses, and one of those is the chickenpox vaccine. So the vaccines now that we have available that are targeting COVID-19 include the mRNA vaccines, and you've all been fully made aware of their roles in vaccinating children and adults. And again, just to recap, the vaccine delivers a message that is the mRNA that is read by certain components of the cellular structures in our bodies that lead to the generation of a spike protein. And the spike protein is expressed on the surface of the cells, and our bodies then generate antibodies to the spike protein. So when the virus comes along with its spike protein, our immune system is ready to attack the spike protein because it has been sensitized, it has been made aware of the need to protect us against the spike protein by having seen it before and develop antibodies and also cellular immunity directed at the spike protein. If you notice that where the nucleus is located is totally separate from where the mRNA is acting. In that regard, the mRNA does not come in contact with DNA. DNA is locked away in a vault in the nucleus. And the example of the peach I show on the slide where the DNA would be locked away in the seed of this peach, and the outer part that you eat, or the fleshy part, is where the mRNA would reside. And that, in terms of analogy, is what happens with where the mRNA is located relative to where DNA. So there's no way that it influences DNA. In terms of the second category of vaccines, the vector vaccines such as AstraZeneca and Johnson & Johnson, the message to create spike protein is actually delivered to the nucleus using a vector, the adenovirus vector that has a key to the nucleus. And when it gets there, it borrows a cellular machinery to make mRNA that leads to the generation of spike protein, and that then leads to the development of antibodies, very similar to what happens with the mRNA vaccine. So in actual fact, the vaccines from the point of where mRNA leads to the generation of spike protein, they're really quite similar from that point onwards in terms of the way the immune system is engaged. Then to summarize, then you have the mRNA vaccines, you have the viral vector vaccines, and then a newer kid on the block, not necessarily newest, but a newer kid, is the Novavax, which is a protein subunit vaccine, involves a synthetic nanoparticle which coats a trimeric spike protein, 3 parts to that, and then there's an adjuvant, and that is delivered to our cells, and again, cells will see the spike protein and will generate an immune response to that. But then there's another vaccine, which is perhaps the newest kid on the block, and that is the nanoparticle, a virus-like particle vaccine. And essentially, this is the Medicago vaccine that was produced, at least the brains behind its production is Canadian, and it essentially uses a plant-based mechanism to generate viral-like particles, where the essentially you have a particle that looks like the COVID virus, but it doesn't have the engine, the nucleic acid component of the COVID virus, SARS-CoV-2. And so it's really just a shell, essentially, with the nanoparticle and the virus-like structure containing spike protein on the surface. And so that then is seen by the immune system, and we respond to that by generating antibodies. So those are the 4 types of vaccines that we have available in Canada. I say that cautiously because the Medicago vaccine has just been approved, but it's not yet available. The one other category of vaccine is where you have the entire viral particle and it's been inactivated. And so there's one of the vaccines that is being looked at in Cuba is one of those, and another in China as well. So what is it that these vaccines are preventing? And I alluded to this earlier. And let's look at the situation with another vaccine, totally unrelated, the chickenpox vaccine. So when the chickenpox vaccine was licensed in 1997, 98 in the United States, it was recognized that there were different outcomes that one wanted to prevent. First, preventing all cases of varicella, any chickenpox case. And you'll see with that, the mean effectiveness was only about 80%. When you look at prevention of moderate and severe chickenpox, it was 96%. And the most severe chickenpox, most severe end of this spectrum, was 99%. And mortality, really, if you got vaccinated, you shouldn't die from chickenpox. And so it's that same concept with the COVID vaccines that we need to get across. And that is when you get vaccinated, you shouldn't have bad outcomes, but you may still get milder forms of COVID. And so with the chickenpox vaccine, another point was that they recognized that earlier on, with one dose, you're seeing these levels of effectiveness, but they found that by giving a second dose, you could actually prevent severe disease 100% of the time. And any disease went up to 98%. With one dose, it was 81%. And this idea then of starting with, one dose, an increase in the number of doses is not unusual in terms of vaccine development and the rolling out of these vaccines, because it's often, only with time that one realizes the true number of doses that one really does need. So in summary then, we have all these vaccines. Now in Canada, we'll likely have 4. These vaccines are best viewed as not necessarily totally focused on preventing any infection, but on moderate and severe infections and also mortality. And the other point, summary point, is that we're seeing playing out in real time the evaluation of the number of doses that are needed. But that's par for the course in vaccine development, where it's not unusual, as is the case with chickenpox vaccine, is you started one dose and in fact you realize you need another dose. And there are other types of vaccines with a similar type of developmental pathway, where initially one thought one needed 1 or 2 doses, subsequently more than those number of doses were needed. So now let's move to some of the questions that just keep coming back. One is, how were these vaccines developed so quickly? And in this regard, it's important to remember that when COVID started, by chance, scientists had been working on another vaccine for viruses that were very similar to the COVID virus, the cousins of the COVID virus. And so one of those was the SARS-1 that came about in 2002, 2004, and that is 80% identical to the COVID virus, SARS-CoV-2. And then the Middle East respiratory syndrome virus, which is 50% identical to SARS-CoV-2. So all of this work was being done before COVID. So when COVID came along, it is perhaps best viewed as luck that this had been done before, because if this work had not been done, and then COVID came in 2019, 2020, it would have taken us another 5 or more years, perhaps, before a COVID vaccine was developed. So we're kind of lucky in that regard. The other luck was that work was already being done on the mRNA vaccine technology for different types of cancers. So all of these two things came together. And then the third lucky point is that when the vaccine clinical trials were done, typically, if one runs into issues, any serious issues relating to safety, then one has to stop the trials, evaluate what's going on, and that might result in the vaccine never re-entering the trial or re-entering 6 months or a year later. And there are vaccines like that. There's a RSV vaccine that was pulled in the 1960s, and since then we've not had an RSV vaccine. So the fact that the COVID vaccines saw the light of day meant essentially that there were no major issues, safety issues along the way. And a likeness to driving to work on a Sunday morning and find that all the lights, all the stop lights are green all the way through. It's an important point for us to bear in mind. So smooth sailing helped getting these vaccines to the market early. The other question that comes up represents a misconception, and that is the whole concept of long-term side effects. Important to recognize that serious side effects that could result in long-term health problems are very unlikely following any vaccine, simply because the period of interaction between the vaccines and the immune system is short-lived, and that's what gives you side effects. And these side effects generally are seen within 6 to 8 weeks after the vaccine. And so it's not usual that you get vaccinated and then all of a sudden a side effect shows up a year later, 5 years later, 10 years later. In the history of vaccine use, there's no scenario like that. Many years ago, there was thought that maybe there was one vaccine, measles, that was associated with a rare condition, but it's now been shown that that's not the case. So vaccines causing long-term side effects that show up for the very first time many years later is really unheard of. And so typically then one monitors for side effects in the 6 to 8 week period after vaccination, when there is interaction between the vaccine and the immune system. The fertility question keeps popping up, and there is no scientific basis to think that there would be a relationship between the vaccines and fertility. Now, that's the scientific background, but what about the real data? Well, there was recently published in January a prospective study from the United States with some Canadian centers being involved, where they looked at the relationship between SARS-CoV-2, COVID infection, and fertility, as well as the relationship between vaccination and fertility. And essentially, they enrolled women who were trying to get pregnant and their partners. And what they showed in essence was that when the male partner had COVID, within, a bit of a typo there, within 60 days of their partner's cycle, this was associated with a short-term decline in fertility. Beyond 60 days, things were fine, but within 60 days of their partner's cycle, there was a decline in the ability to get pregnant. Only when the men were infected. When the women were infected, they didn't see that drop in fertility. When they looked at the vaccines, they found that vaccination itself did not affect fertility when either male or female was vaccinated. So the key point there is that the vaccines did not affect fertility. What was affected fertility was COVID infection in the men. Fortunately, it was short-lived. Now, what about the third doses of the mRNA vaccines? And like all of you, I had the same questions that I asked myself. And the first is, do third doses provide greater effectiveness? That's the first question. And here are data from the United States that look at 2-dose and 3-dose regimens. They study mRNA vaccines. And again, here we're targeting the severe end of the illness spectrum. In other words, what is probably more important, hospitalization or worse. And so in the blue column would be where 2 doses are used, and in the yellow column, 3 doses. First green row, where people were with normal immune systems, and the second would be where they had abnormal immune systems that were immunocompromised, so-called weak immune systems. And essentially, what it showed in terms of the vaccine effectiveness, in terms of preventing hospitalizations, with 2 doses, you had an 82% vaccine effectiveness. But for the people with weakened immune system, it fell down to just less than 70%. However, when people got their third dose, you see where the vaccine effectiveness rose to 97%. And even when you have a weakened immune system, it got up to just less than 90%. So it really showed that a third dose, at least in adults, was associated with an incremental gain in effectiveness over and top of the 2 doses. So that's the first question. Now, we know that there are differences in effectiveness based on variants. And so during Omicron predominance, what they found was that you had a 2-dose regimen with effectiveness of 81%. But when you got to the point of individuals who received a third dose, you had an effectiveness that was well up to 90%. And so with Omicron, it got up to 90%. The previous slide, if you notice, it was August to December 2021. And so this would predominantly be Delta. But with Omicron, it was also beneficial in preventing hospitalization. Now, when we look at vaccine effectiveness among adolescents, what do we see? So individuals 12 to 17 years of age, period would be July to December of last year. And what are we seeing? Well, what it basically shows was that for individuals who were fully vaccinated with 2 doses of the vaccine, you had pretty solid vaccine effectiveness, which is comparable to what one sees with the adult data that I showed you earlier on. When one looks at the ability of the vaccines to prevent symptomatic infection, it shows the effectiveness then in different age groups. And essentially, the key point was that in preventing symptomatic infections, you had effectiveness that was in the high 80s up to high 90s, but it varied based on age groups. You can see that the group with the highest vaccine effectiveness was the 12 to 15 age group. Then we have the 16 to 19, and then we have adults. And the second take-home point from this is that if you look at trends over time, you can see where the effectiveness wanes over time. And so by the time you are at 180 days, you can see that your effectiveness has dropped below 70%, which is not bad, but it certainly isn't ideal. And this is, again, directed at symptomatic infection. So in an ideal world, then, one wants to introduce a 3rd dose to prevent bad consequences from the waning immunity that one sees. And so if you look at the effectiveness of the vaccines then in individuals who are age 5 to 11 and 12 to 17, there was one study that came out of New York that was widely reported, and it caused some concern among families regarding, well, gee, you know, is the vaccine really effective in these young children? And so this study looked at over about 850,000 individuals, 12 to 17 years of age, and then 365, 5 to 11 years of age. What they found was that the 2-dose vaccine protection against infection for the younger group dropped from 68% to 12%. But in terms of hospitalization, the effectiveness fell from 100% to 48%. With respect to the older age group, one sees protection against infection dropping from 66% to 51%, and hospitalization 85 to 73%. So that's not really great. One sees a drop in protection. Now, before we had a COVID vaccine, if you recall, when experts were asked what sort of effectiveness they would be, happy with, there are many experts around the world, including Anthony Fauci, had said, well, I don't know what we're going to have, but if we have something that's about 50% effective, we'll take it. But we came in with vaccines that were showing, at least in the efficacy studies, more than 90% efficacy rate. And in real world effectiveness, I was pretty close to that, as you saw with the previous slides. But then when we end up with situations where reports suggest you have rates now that are close to 50 over time, then people wonder, are these vaccines doing what they're supposed to do? So just a matter of putting things in perspective. That said, the CDC looked at this more closely. In this regard, they looked at hospitalization during Delta and Omicron predominance. We have 3 different age groups, 5 to 11, 12 to 15, and 16 to 17. And what the CDC data showed was the 5 to 11 effectiveness of 74%, for the 12 to 15, 73%, and for the 16 to 17, 94%. So effectiveness between 73 and 94%. And that is still pretty decent. That's still pretty good. As I said, you know, it depends what one's expectations are, but 73 to 94% is still quite good. So overall, I think we have good data to support continued vaccination of kids with the current vaccines, and an ideal world where the boosters have been approved for the particular age group, there's advantage in receiving a third dose. The question is, though, are these third doses safe for teenagers? And that's a really important question. And just a bit of a backdrop of why we're thinking about this is that the risk signal that has emerged with third doses is myocarditis and pericarditis. And it has been shown that the myocarditis and pericarditis curves tend to mirror what we see in real life prior to the COVID vaccines, when myocarditis and pericarditis are occurring across different age groups. And so the bottom line is, as you get younger, the myocarditis signal falls. The 5 to 11 age group we predicted would be a quiet zone for myocarditis, and that's exactly what has happened with the vaccines. It's a quiet zone for myocarditis. As you get below age 3, other factors contribute to the occurrence of myocarditis, and you might get a kind of a mini U curve, so high, low, and then it picks up again a little bit as you get below 3. But the 5 to 11 is a quiet zone for myocarditis, which is good. So the group above 12 is a hotter zone for myocarditis, if you will. And so you can imagine that if one is talking about third doses, you want to make sure that the third dose was not going to bring about more myocarditis than the first or second dose. We know that the second dose results in more myocarditis than the first dose. Overall myocarditis is rare, but it does occur. And the second dose sees more cases occurring after the second dose than after the first. So what about the third dose? And so the key point is that when they looked at individuals who received a third dose, and these are data coming out of of Israel, and there are some data coming out of the UK as well, is that among more than 6,000 individuals, teenagers 12 to 15 who received a third dose, there were no cases of myocarditis reported. And guess what? It's all early, but it's an important signal that it does appear, because there are some data from the UK as well, that after a third dose, the myocarditis signal seems to be lower than what was seen after the second dose. So this is really, not only a good news story, a great news story, essentially saying that after the third dose, for whatever reason, the myocarditis signal is lower. So if you just follow the cursor where the red box is, you can see myocarditis, a signal after the second dose, nothing after the third dose in the 12 to 15 age group, females, and more cases in males, so we know that. Again, nothing after the third dose. And so in summary then, the key points is that the data that are emerging would suggest that the risk of myocarditis of the third dose is not higher than after the second. It's rare after the second. It's even more rare after the third, perhaps even lower than after the third dose. And then if it does occur, key point is that it tends to be rare, very mild, kids recover without consequences. Now, perhaps one way to look at this issue is that if teenagers did not have myocarditis after the second dose, it is highly unlikely that they would have it after the third dose. So that's perhaps the take-home point. What about the timing of the third doses? Now, individuals in Ontario who are aged 12 to 17 years of age are eligible to receive booster doses at equal to or greater than 6 months after completion of their primary series. And we know that there's been some discussion around the country as to, is this appropriate? These are the recommendations in Ontario. Individuals could get a third dose earlier based on their particular risk profile. Now, to summarize the rationale and the Ontario recommendations, individuals who are 12 to 29, the recommended vaccine is the Pfizer vaccine because there are lower rates of myocarditis, pericarditis following the use of the Pfizer vaccine compared with the Moderna vaccine. So if you're under 30, you get Pfizer. If you're 70 and above, you can get either Pfizer or Moderna. There are some data that suggests that in this age group, the Moderna vaccine, the higher dose, may result in a better immune response. For individuals who are not 70 and above, who are not long-term care facility residents, who are not immunocompromised, for all these other individuals, they can get either Moderna or Pfizer. The Moderna, when used in these other individuals, would be the 50 micrograms as opposed to the higher dose in the adults of 100 micrograms. And so to bring it all to an end, to summarize where we are now, that one is that with the third doses in children and adults over the age of 20, the third doses are safe, number one. Number two, we know that immunity after 2 doses will wane with time, as you can see on this graph. And the third dose does provide additional protection. For the approved age groups, underlined approved age groups, the benefits of the third doses do outweigh risks. And so we do recommend that people get their third doses. Now, you've noticed that, at some places I said booster, and towards the end I speak more of third doses, because it might well be that at the end of the day, the primary series, the package of doses that we need for proper vaccination is actually 3 as opposed to 2 with the mRNA vaccines. And so 3 doses, the third dose might not necessarily be a booster dose. It might just be part of what we need. But it's clear that there are some people who will need it more than others based on the competency of their immune system. And so with that, I will stop so that we have some time for questioning. Thank you so much.
Thank you very much, Upton, for very, very elucidating presentation, particularly focused on pediatric vaccines. So I invite you to post your questions in the Q&A, just so that we'll be able to answer them as you post them. That's the first question. You talk about 3 doses rather than booster language. How likely is 4, 5, 6, etc.? How can we communicate this with the public in a way that they buy? I mean, would they not run out of patience with the numbers?
Yeah, that's a great question, Isaac. I think that the idea of a 3-dose primary series, I think is one that can be sold, and public will accept that if individuals are at risk. Once you start to get into a 4-dose primary series, that becomes an issue. So I think the role of the 4th dose is really, truly, in my opinion, best going to be defined and messaged as a booster. And the question then is, like with any scenario in infectious diseases where you provide a booster, you boost for 2 reasons. Number one is that a person is at high risk. And that means that if they got infected in a setting where immunity might be waning, their outcome would be devastating. An example would be people in long-term care facilities, or the immunocompromised, lung transplant patient, the cancer patient on therapy. The second reason would be the person is not likely to have the best sustainable immune response after the primary series of 3 doses. And so for that individual, you might want to provide a booster. And another reason might be if we have to put out a fire again, and let's say we had Omicron version 2, then one may look at whether or not a booster is needed, and should everyone get the booster or only selected individuals. So the question then, though, is, let's say in the fall of 2022, we have the necessity for selective boosting of individuals. Would we also need to do the same for the fall of 2023? And that is a possibility. But as we move forward in time, I think that it's likely that the groups that end up being boosted would be more clearly defined. It may be, it is going to be such that as we move forward in time, Isaac, the folks who get boosted are residents of long-term care facilities or people with immunocompromising conditions.
Right. So, Upton, in terms of the language, are there any primary pediatric vaccines that require 3 doses as primary immunization? Perhaps giving those examples will make people realize that this is not new.
Yeah, I think that's a really great question. So if you look at the childhood vaccines, like the pneumococcal vaccines, and when a kid first gets their vaccine, two months, then another 2 months, and then another 2 months. And then we have the chickenpox vaccine. I made reference to that earlier, where typically people don't need a third dose beyond their 2 doses, but occasionally we need to give an additional dose because somebody has a weakened immune system. People going into transplantation, for example, some individuals who are going on treatment because they have rheumatologic conditions that require, medications that alter their immune system, and you check their chickenpox titers, you realize that their response is not great, and you give them another dose. And then the hepatitis vaccines, which 3 or 4 doses depending on the scenario. So there are examples like that, and it's just that what we're seeing happen right now is that the public is seeing the determination, the process of determining the number of doses play out in real time. But in fact, I often say, if you had fallen asleep at the start of the vaccination campaign, and then you woke up at the end of the vaccination campaign and you're told that it's 3 doses, you'd say, oh, okay, I'm good. It's just that you're seeing it play out, along the way. And it sounds like uncertainty, but it's all part of the evaluation process.
Are there any strategies that we know already that may prolong the level of neutralizing antibodies for longer, like adjuvanted protein conjugated or other means in which one could prolong? Because people are thinking, if this is going to be like the flu, then does it mean that every year we will need a vaccine? Or could it be that this interval will be a little longer?
Yeah, that's a really great question. We don't have that information yet, but what we do know is that there's some evidence that's emerging that suggests that that the shorter intervals between the doses might be associated with a lower type of immune response compared with a longer interval. So that is something. With respect to the roles of adjuvants and other things, we're not there yet. But definitely, though, there's studies that are being done, including ones that we're doing, that you're aware of, Isaac, that will look at the sustainability of immune responses across different groups of individuals to see whether or not they're the same. Like, for example, if you're East Asian and you get vaccinated, does your immune response 6-9 months later sits at the same level as somebody who's of African or European ancestry? These are data that we have assumed, but might not necessarily be the same based on other data relating to other types of vaccines that have been studied previously.
Another questioner is wondering why we focus solely on vaccine and not on progress being made with therapeutics. What's your thought about that? Should there be a combination of both talking about vaccine and therapeutics, or you think that's a wrong strategy?
No, I don't think. I think it's the talk today is on vaccines, but we could certainly have given a talk on therapeutics. But so it's not an either or situation. The presentation today is on vaccines. But that said, it's important to recognize what therapeutics do and what they don't do, and that therapeutics target treatment at the point where somebody has been infected for the most part. I'll come back to prevention in a minute. And so what that does is that it will potentially prevent severe outcomes from infection and disease. But the way that our immune systems work is that once you get infected, you may get the consequences of infection even if you don't have severe disease. And so we have examples of that in infectious diseases. A good example of that, as you know, Isaac, is cytomegalovirus infection in patients with weakened immune systems, that you get infected and CMV, cytomegalovirus, starts to play with your immune systems and can give you all kinds of different things, even if you don't get disease. And the treatment focuses on the disease part of it. So the therapeutics are important, but they have a specific role to play, and I think that's important to recognize. And the moment the strategies for rolling out therapeutics and how to use them in a way that they're most beneficial is still being sorted out, where for therapeutics to be most effective in an ideal world, you want to get them on board right at the get-go once infection has occurred, and that is hard. And so you're looking at the very most 3 to 5 days after symptoms. But it is certainly a different type of presentation than a presentation on vaccines, but not to ignore the potential role of therapeutics once we have that sort of data. We have some already, but there's more to come. Another point I should mention about therapeutics is that, because it's just something that we didn't really, again, one of those things we didn't articulate very clearly to the public. And that is in the history of the treatment of respiratory viral infections, we have no medications that are curative. And that is partly because of what I said earlier, in that at the point where we start to treat, the horse has already left the gates. Yeah. And so we kind of just trying our best to slow the horse down, so to speak. And the best example we have would be the treatment of influenza, where, you know, oseltamivir and those types of medications just help to shorten the duration of illness. There may not be a sort of night versus day type of difference in terms of impact on outcomes.
That's great. That's great, that perspective. So, Upton, based on what we've learned with the rollout of this first generation of COVID vaccines, if the scenario were to play out again in a year's time, what do you predict will be a better approach from the lessons we've learned? For example, the ideal interval between doses, the availability of early diagnostic sort of antigen-based maybe diagnostics to be able to quarantine and even initiate therapeutics. Are there key lessons we've learned in this rollout, given we were in the dark, that informs best practices going forward, from your point of view?
No, that's a really great point. I think it's not so much the lessons learned. Maybe one of the lessons learned might be that there's some flexibility in the intervals between the vaccine doses. Right. I think that's one that we've learned, and clearly we know that multiple doses, at least for the current vaccines, more than one dose is beneficial than a single dose. And so chances are, if you're trying to put out a fire, you might need to use a shorter interval because the fire is burning then. But if there isn't a fire to put out, then you have time, perhaps a longer interval. But I think the other thing that we've learned is really more a matter of the development that has resulted from the availability of at least 6 different vaccines, of which I think the main focus is going to be in Canada, will be on 4. So the 6 that I'm talking about are Pfizer, Moderna, AstraZeneca, Johnson & Johnson, and then Novavax, which is new, and the Medicago vaccine. And so perhaps a better understanding of where mixing and matching might occur is something that we are learning, and for next year we would be in a better place. So, for example, in the rare scenario where kids have gotten myocarditis after their first or second dose of an mRNA vaccine, if they need to have a third dose, should that then be Novavax or Medicago? And that would be an advancement in the field, so to speak.
Right. No, that's very, very helpful. And do you think, just a final question, do you think, given what we know about Omicron and the evolution, that the virus really seemed to have evolved into higher transmissibility and a lower level of virulence? Is in the evolution of other viruses like the flu virus, is that the trend, that the evolution of these viruses occurs over time?
Yeah, that's a great question, and the answer is yes. And the way to think about it is that the way viruses need living cells to replicate and to survive. And a virus that kills off its cells and kills off its host, in other words, is severe, is also killing off itself. It's suicidal. And I used to say that a stupid virus is like HIV, you know, but viruses that are easily transmitted, that cause mild illness, or viruses that tend to evolve and we live with, and they tend to go on and keep going on. And so I think that's a natural evolution in terms of what we expect, that you'll get more transmissibility but milder illness as you move forward in time.
On that note, I think we will bring it to a close. It looks slightly more hopeful, and now we have at least 6 types of vaccines which will be available to Canadians. Thank you very much, Dr. Upton, Alan, for this very, very interesting and elucidative talk. So we ask you to give your comments and your assessment of these programs, because they help us in shaping how we design them in the future. Thank you everyone for attending, and thanks Dr. Allen again for this talk.
Thank you very much.
Bye-bye.